but before we do that, I want to kind of get finished through this set of notes, basically covering the rest of different sort of basic animal systems. Animals accomplish basic problems, so we looked at some feeding, digestion, and things like that. Now I want to talk a little bit about internal transport. So if you're very small, you can just rely on diffusion, but that kind of limits things. And so as animals get larger, they need to be able to distribute nutrients to the various cells. They need to be able to deliver oxygen to the tissues, pick up waste like carbon dioxide, ammonia, and so on. And so they need to have some sort of an internal transport system. The simplest form is, as shown in these diagrams here, is sponges and mammalians. You don't have actually a true internal transport system. You basically just use the cavities, the sponge or seal, or the various channels that run through the sponge. Allows most of the tissues to be in close proximity to the external environment. You can circulate some nutrients through here. For cnidarians, which you'll meet in lab today, again, you have a large gut, a gastrovascular cavity with a single mouth opening here. So it's an incomplete digestive tract. But again, all of the cells are either fairly close to the external environment, so they can just get gases and things by diffusion, or fairly close to the gut cavity lining these internal cavities here. But again, all of these animals have relatively simple organization. In particular, they only have tissue level, don't have organ level. They don't have dense packs of tissue all the way from the surfaces. Most of these are just lined, sort of single cells here, layers that line the internal and external environment. All right. So for larger organisms, they need some sort of a true internal system, not just a gut. And so you have true circulatory systems. Circulatory systems contain a fluid, just called either blood or hemolymph, depending on the kind of circulatory system. We'll get to that in a minute. And that fluid is then contained in a combination of vessels and or chambers within the body that is not open to the external environment. And very commonly, circulatory systems also have very fine vessels with very thin walls that deliver fluids and food and nutrients and gases and so on directly to the tissues. And so we call those fine cells or capillaries, those fine vessels or capillaries that can branch out and get in close proximity to most of the tissue, so you don't have to rely on much diffusion. And again, they have often very thin walls to them, so that, again, there's not a lot of distance for nutrients and gases to have to diffuse across. So that's, again, capillary beds are a common feature of most circulatory systems. The fluid then itself can be moved through the body, either just through the basic body movements and contractions, so that if you have a large fluid-filled cavity or even vessels, and you bend your body, you squeeze on one side, that's going to push the fluid in one direction, bend the other way, and it'll move in the other. So simple body contractions, especially if it's coupled with a hydrostatic skeleton, where you have internal pressure, can move fluids around, and that may be all you need. Again, if you're larger and more complex, you may want to have a blood movement of those circulatory fluids independent of your body movements, in which case you'll need blood vessels that become larger, thicker, and muscularized, and act as pumps, in other words, hearts essentially. So hearts are really just muscular blood vessels. In some cases, like ours, very complex, but lots of invertebrates are just simpler. They're just kind of a swollen blood vessel surrounded by muscle. All right, so the two main types of circulatory systems are open versus closed. A closed circulatory system is what we have, where the fluid, called blood, so blood is basically associated with closed circulatory systems, is contained entirely within a system of vessels. We do have, there may be extracellular fluids as well. You still have a sealant, for example, in most cases. And so that you have your coelomic fluids, your interstitial fluids between cells, but that's separate to a large extent from the fluid that circulates within the vessels. And that fluid may be chemically and even cellularly different from the other kinds of fluids. And so we typically find closed, so here's a very schematic view. you have a sort of worm-like animal, mouth at one end, the gut goes through, anus at the other. The dark black here represents the circulatory systems. You have, again, going into capillary beds, you have capillary beds. But all of the fluid stays entirely within the system here. You can have thickened areas where you might have hearts, where they're muscular, and it's pump the blood, and so on. In general, closed circulatory systems are found in animals with a well-developed true coelom. Can be often subdivided. In contrast to a closed circulatory system, an open circulatory system is just that. The vessels don't form a sealed system, but instead are open not to the external environment, but to the other fluid-filled cavities, to the coelom or pseudocoelom, depending on what the animal has. And so the fluid in open circulatory systems is called the hemolymph, rather than blood. And so the black here represents, again, the blood vessels. You can have a pumping structure, but there are openings here where fluid can enter into the heart, and the vessels themselves open up into the hemocoel. So the fluid spends part of its time in the circulatory system, but it also moves just through this larger hemocoel cavity as well. The hemocoel is going to be either basically derived from the coelom or a pseudocoelom, depending on the animal. So very often the hemocoel may have other functions, like acting as a hydrostatic skeleton, in addition to acting as part of the circulatory system. So examples of closed circulatory systems include us vertebrates, for example, and octopi and squid have closed circulatory systems. Annelid worms have closed circulatory systems. Open circulatory systems are found in most other mollusks besides squid, and in arthropods generally have an open circulatory system, and so on. All right. So one of the even if you're moving fluids around, one of the things you want to do is deliver oxygen and get rid of carbon dioxide. The problem is that oxygen is a nonpolar molecule, and so it does not dissolve terribly well in water. The oxygen levels in water tend to be much lower than in the air, for example. And the warmer the water, the less oxygen it holds. So if you have a relatively warm body or live in a warm environment, that makes it even tougher. So there is some oxygen dissolved, but you need to circulate a lot of fluid if the organ is relatively active and therefore has high energy and oxygen demand. So one way to improve the ability of your fluid to carry oxygen is to use a respiratory pigment. A respiratory pigment is a molecule that binds to oxygen and therefore increases the ability of that fluid or the blood or the hemolymph to carry the oxygen. And they're called pigments because they do tend to happen to often have, in order to effectively bind oxygen, they usually have a metal ion associated with them, and those tend to impart a color to them. The color is sort of a secondary effect of how they work. So the one that you should all be familiar with is hemoglobin. That's, of course, the same respiratory pigment that we use, and it has an iron atom in its center, and that iron atom gives the hemoglobin a red color. The brightness of the red basically determines whether or not it's bound to oxygen at the time. And this is actually probably the most common respiratory pigment. I should point out that in vertebrates, the hemoglobin is carried in specialized cells. It's packed into red blood cells. That's not true for the vast majority of invertebrates. The hemoglobin would just be a protein floating around in the fluid, not associated with specific cells. There are some other respiratory pigments. The second most common is hemocyanin, and that has copper as a base rather than iron, and that imparts a somewhat bluish color, although it's usually don't find it that are very bright blue. Hemocyanin is found primarily in things like mollusks and arthropods. There are actually some other respiratory pigments as well, beyond those two, but they're fairly rare and we're not going to worry about them. These are the sort of big two. For animals that use respiratory pigments, it's usually either hemoglobin or hemocyanin. All right. So that's circulatory systems. One of the things we want to do is, of course, use that to carry oxygen and carbon dioxide, which means you have to be able to exchange carbon dioxide and oxygen with the environment. Again, if you're relatively small, you can just use any kind of permeable, you can just use your skin for gas exchange. So any sort of permeable surface that allows water to pass through will also allow oxygen and carbon dioxide dissolved in that water to pass through quite easily. So it's generally fairly easy to carry out gas exchange. And for smaller animals, and certainly protists, the surface of the animal is sufficiently large relative to their needs that's primarily how they carry out respiration. It's primarily cutaneous. You might have secondary absorption of gases, exchange of gases through the gut, and so on. But as other animals get larger, remember, as you increase in size, your need for oxygen is largely proportional to your mass or your volume. That's going to increase as a cube of your size increase. The surface area only increases as a square, and so the surface area increases more slowly, so large animals have proportionally less surface area. So it makes it much more difficult to simply rely on cutaneous respiration, just gas exchange through the skin surfaces. So that means that larger animals basically need specialized structures whose primarily work by having a shape that greatly increases the surface area across which gas exchange can take place. All right, so give just some general examples here. So those parts can be gas exchange parts can be internal or external. So for example, in sea cucumbers, they basically can breathe through their butts. So this is a sea cucumber, mouth is up here, anus at the other end. The digestive tract, this is the gut right here, and then it kind of enters into this hindgut part here, and then poops out the anus. There are branches off of that hindgut called respiratory trees. They're basically they just branch finer and finer so that they have lots and lots of surface area. Then there are blood then vascularized around it. There's fluids around that respiratory tree that can pick up that can carry out gas exchange. And they can actually, the hindgut is muscular, so they can pump water in and out of their anus, basically, in order to breathe. And so it's an expansion of the gut. We really could, they are kind of like any other gill, basically, but except that now they're not really quite like gills because of fluids on the inside. All right, so gills is a fairly general term. You'll see it used in a lot of organisms. Don't assume necessarily that gills are homologous. They've evolved multiple times in different organisms. The term is basically used for any sort of mostly external, can be somewhat protected, but largely exposed to the external environment structure that's used for gas exchange. And those structures are usually highly folded or convoluted. They may be made up of lots of little tubes or little flat sheets or plates or something like that. But those all of those foldings create lots and lots of surface area across which gas exchange can take place. So here, for example, is a sea slug called a white-spotted sea goddess. And you can see that, so you have the yellow areas of skin, it has some sensory structures at its front end, a big muscular foot down that you can see sticking on the back. And then this fuzzy stuff over here, you can see it's kind of got lots and lots of folds, even at the microscopic level. These are sort of external gills on the back. Now, it turns out if you look at snails also have gills, but they actually have different kinds of gills located closer to the head. And those are not homologous to these sort of secondarily derived gills in some of the sea slugs. But they work the same way. They're basically highly complex, highly convoluted structures that have lots of surface area. All right. And so in marine organisms, if they have a respiratory structure, there are going to be a few internal things like the sea cucumbers, but most organisms are going to have something like gills. They are more or less external, and here they're very obvious and external. In other cases, they may be under some sort of a little protective covering, but they're still basically directly exposed to the seawater. And the inside of the gills is going to be where the blood or hemolymph flows to pick up gases. Here, the blood and hemolymph is essentially on the outside of the structure of the internal structure. All right. If you're carrying out gas exchange in the air, however, it becomes a very different scenario. Air is much different. One advantage of air is that the oxygen levels are much higher than they are in water. The atmosphere is about 22% oxygen. Most water is going to, the oxygen levels in water are going to be, the dissolved oxygen levels can be quite, percent or less or something like that. Very much, so much higher concentrations of oxygen. But you run into physical problems. One is that most gills, if you look at regular gills, they are very fine, delicate structures so that you can maximize surface area without a lot of tissue. And so you take those same delicate structures that are supported by the natural buoyancy of water, they have about the same density of water, and you stick them in the air, they're just going to collapse on each other. They're just going to go flattened, and you lose all of that surface area. In addition, if your gills are exposed to the air, remember, in order for gas exchange to take place, they have to be wet. And so, you'll have to have, for gas to be able to pass through, water also has to be passed through. So that means that exposed gills will dry out and can dry out the whole organism very, very quickly. So that means, sort of typical gills that work well in water do not work on land at all, despite the higher oxygen concentration. So, what that means is that respiratory structures for animals, for terrestrial animals, for structures that work in the air, generally have to be internal. They have to be moved on the inside, and that way you can minimize the sort of opening, the amount of that's exposed to the external environment. You can have small little holes, like our nostrils and our mouth are relatively small compared to the surface area of our lungs. And so we minimize the amount of moisture that's lost. You're going to lose some, but you minimize that. And you want to have a structure that doesn't require the natural buoyancy of water either. So you need to change those internal structures. For invertebrates, there are three basic air-breathing respiratory structures. There are book lungs, regular lungs, and trachea. Let me see, because I don't think you can see that. Let me make it a little darker here for a moment, sorry. So what I wanted to show you here with this slug picture, this is a banana slug. So it's a terrestrial mollusk, so it has to breathe. This part over here is a mantle that basically covers most of its organs. It's kind of a skin-like structure. But you can see right here, there's actually a hole here. That opening is the opening to its lung. So it has a lung that basically sits underneath the mantle. It's basically right in here. On top of the organs, there's this basic sac. That the surface of that sac is vascularized, so there's lots of, in this case, hemolymph that would be near that sac. But it has a relatively small opening through which air moves in and out. That works pretty well because it's easier to move air than it is water. Because water is the air is much less viscous. Remember that. And so you can move air in and out fairly easily without needing a large opening. And you have a fairly small opening that doesn't lose a lot of moisture. And you can even close that off, basically hold your breath, if you don't need too much oxygen and therefore conserve more water. So that's an example of a lung. It's an internal sac that sits about here and open to the environment. In some arthropods, you have things like book lungs. Book lungs are basically a infolding of the external skin, the epithelium, into a series of flat plates that are mostly covered on the to the outside. So there's relatively small external opening, a series of flat plates underneath the skin. And they're called book lungs because those plates look kind of like pages of a book. So book lungs and lungs work on a similar principle. That is, you have a relatively moist cavity across which respiration can take place. The final system, found primarily in various arthropods that are terrestrial, is a tracheal system. And so a tracheal system is basically a series of air tubes that bring the air directly to the tissue. So in lungs and in book lungs, the respiratory structure is internal, but it's only in part of the animal. So the lung is basically sitting right here on the snail, and then the oxygen that's picked up has to be circulated by the hemolymph to the head and then the tail and so on, the back end and all of that, all of the rest of the animal. The same is true with book lungs. They're often on the ventral side of certain animals. And then the oxygen has to be picked up from the book lungs and delivered. Trachea work a little bit different. They don't actually require much of the circulatory system because they are, so here on the right side is the open outside and external environment. The cuticle would be the waterproof covering around the skin here, the epithelial cells of the outer layer of skin. And there's an opening here. The tube folds inwards. And this is filled with air here, so it's just directly opening to the air. The spiracle, this can often be open and closed, so again, you can take a breath. And then the trachea branches into smaller and smaller branches, eventually into tracheals, bringing the air into the actual tissues. And so the tubes themselves branch throughout the body, so there are multiple openings, spiracles throughout the animal, and they each branch and basically deliver air directly to all of the tissues. No circulatory system needed in that particular case. Okay. All right. So moving right along here, next system is the final system is the nervous system, which includes the neurons and sensory organs. So nervous systems are basically composed of specialized cells that conduct electrical impulses. They are found, nervous tissue is found in all animals except for sponges and placozoans, and perhaps a few weird parasitic forms that we'll worry about later. And so even things, relatively primitive animals like cnidarians and ctenophores have a nervous system. They have so specialized nerve cells is one of the kinds of tissues that is found in these animals. In most radially symmetric animals, they have what's called a nerve net. That is, there's a net-like arrangement of nerves. It's all of these little lines here represent the nerves here in something like a sea anemone. It is not centralized. There is no central brain. There's often very little concentration of neurons. A ganglion, a cluster of neurons. They're just kind of diffuse. And in fact, in cnidarians, the electrical impulses, unlike in our nerves, which are unidirectional, our nerves conduct electricity only in one direction. That's not the case in cnidarians. They can actually often conduct electrical impulses in both directions. So something, say, grabbing a tentacle on this side here would stimulate a bunch of impulses to be sent out. It would be causing some of the other nearby ones to basically respond to that signal, because some of those signals would reach the muscles and cause them perhaps to pull the muscles back. And it would be diffuse and take longer and be slower and less. The signal would be much stronger when it gets to the other side of the animal. So, only the tentacles on the side where it gets, let's say, nibbled on would actually retract or respond. So this is a diffuse system, and it's typical of radially symmetric animals. On the other hand, most bilaterally symmetric animals tend to have a somewhat more complex system that is divided up into a central nervous system and a peripheral nervous system. So here, for example, is a typical nerve ladder of a flatworm. And so it has a central nervous system with one or more nerve cords, main sort of a bundle of nerves that run along the length of the animal. In this case, you can see there are two main longitudinal nerve cords here. And then at the head end, they may often have enlargements. That is, there are clusters of nerve cells here that are processing information. Because if you have a head, what makes a head is the fact that you have a bunch of sensory organs around there. That's the direction you move, and you want to generally know where you're going more than where you've been. So you're going to put most of your sensory organs at the front end, at the anterior end. And so then you need to have a bunch of nerves that process that information, which can now get more complex, and send it out to the muscles along the way. So you'll have ganglia. Ganglia are basically enlargements of nerve cords, concentrations of neurons that can process things. And, at some point the ganglia get large enough where we can call them the brain. There's no real hard distinction between what is just a ganglion and what is a brain, at least in animals like this. So you could, I suppose, call this a kind of a brain, relatively small, simple brain, but the brain nonetheless. So the central nervous system is responsible for sort of coordination of activity and overall control. And then the peripheral nervous system are all of the nerves that connect from the central nervous system, from the main nerve cords out to the muscles and organs, and also from sensory receptors back into the central nervous system. And this is typical for most bilaterally symmetric animals, but there are a few exceptions. So those acial flatworms you saw, for example, are bilaterally symmetric, but they still have only a relatively simple nerve net rather than any kind of true nervous system, any kind of a central nervous system. All right, and then finally, I want to just kind of go through some of the major types of sensory structures and sensory organs that animals have. And so again, just a lot of this is just really a lot of terminology here. So tactile receptors are touch receptors essentially. They detect contact with the external environment in various ways. They are often in animals associated with things like bumps in the epithelium. Little bumps and spines may often be loaded with tactile receptors. Setae, bristles, and hair-like structures will have them as well. They seem to be pretty close to universal among animals to have some sort of tactile receptors. And the next kind of receptor is called a georeceptor, and it responds geo for Earth. It detects which direction is the Earth. It basically detects the pull of gravity, and so tells the organism which way is down. A typical georeceptor looks something like this. This is a statocyst. It's a kind of organ that is it's a kind of georeceptor. So georeceptor is just a functional term of what it does. A statocyst is a specific kind of georeceptor that's fairly common, and it's basically a sphere of cells that form a hollow sphere that is filled with a fluid, and inside that fluid is a statolith. Lith means rock. Basically, this is usually a little mineralized bit of something, a little bit of calcium carbonate that's crystallized inside that fluid, and it's denser than the fluid, so it tends to go down. And surrounding that fluid-filled sphere are the sensory epithelia with little cilia that detect when the statolith bumps against them and displaces them. So which of these cells basically get stimulated tells the animal which side is down. Another kind of cell we don't have pictures of here are proprioceptors. Proprioceptors, again, are probably pretty universal among animals. They're strictly internal receptors, that is, they don't respond to external stimuli, but respond to internal stimuli. They detect the positions of body parts, the amount of tension and stretch and pressure on body parts, and kind of tell the animal what its own self is basically doing, its body positions and so on. And so, again, proprioceptors in more complex animals like humans can take a wide variety of forms, but a lot of them are fairly simple. They may just be simple exposed nerves that, if you stretch them, they trigger an action potential, that kind of thing. Another kind of receptor are phonoreceptors, which detect sounds. Sound receptors are actually pretty uncommon in invertebrates, in part because it's actually fairly tricky to detect sounds, not impossible, but it's relatively tricky to detect sound in a marine environment because organisms are pretty much the same density as the water, and so sound waves simply pass through. You need something that's sort of at a different density that can trigger appropriate vibrations rather than just passing through. And so for marine organisms in general, phonoreceptors are uncommon. They're not unheard of. And there are even some shrimp and so on that actually communicate using sound. But it's fairly not terribly common except for some terrestrial arthropods. So there are moths that have phonoreceptors that can hear the calls of bats. There are various crickets that can hear the calls of their potential mates. There are praying mantids that hear the calls of crickets and find their food that way. So for terrestrial animals, phonoreceptors have evolved a number of times, and it can be in lots of different places. A baroreceptor is, so think of barometer, measures pressure, overall pressure. They particularly changes in pressure. So they can tell, and changes in pressure help you determine what depth you are in the water. That appears to be important for vertically migrating species. They can also change, sometimes detect changes in pressure that might to help detect particular kinds of ocean currents and weather changes and things like that as well. For the most part, we don't know. There have been experimental studies that show that organisms respond to changes in pressure, so they can detect it, but we don't actually know how they do that. We haven't identified the receptors responsible yet. Once again, the next receptors are thermoreceptors. As the name kind of so suggests, they detect temperature, particularly, again, changes in temperature. So they can be useful for detecting the right microclimates, seeking out warmer, cooler places. Some species actually can use it for detecting food. So leeches, for example, look for warm bodies as a source of a blood meal, for example. So they have actually, not only can they detect changes overall, but they can detect to some extent the direction which they're coming from. Once again, we don't know how they do that. In vertebrates, for example, thermoreceptors appear to be just sort of bare nerves. They don't seem to be associated with any kind of specific structure, and that's probably the case with invertebrates as well. There's some specialized nerve that doesn't necessarily look particularly different from any other nerve. Then another type of receptor are chemoreceptors. Basically, this is smell and taste. The ability to detect chemicals in the environment. This is probably pretty much universal in invertebrates and probably protozoans as well. You need to be able to, detect, food sources, potentially dangerous chemicals, areas with low or high oxygen, and maybe all kinds of things you want to detect in your environment. Very commonly, chemoreceptors can be found anywhere in the organism. Again, the head is more likely. If you're a bilaterally symmetric animal, you're more likely to have those receptors on the front. But they can be kind of anywhere. They can be. They're commonly found in pits and depressions in the body. So if you look under a scanning electron microscope and see little pits, that's a good chance that it might be chemoreceptors at the bottom of that pit, picking up chemicals. They often also may be associated with setae, bristles, or hairs, hair-like structures, that can also be lined with chemoreceptors as well. And not surprisingly, there are a huge variety of forms. Some have obvious external structures, some are much simpler. The capabilities of chemoreception varies quite a bit from among different organisms. And so there's lots of variation that we're not going to get into right now. We'll occasionally see some later on when we look at some of the groups. All right, and our final, I keep saying final, I think, but this is going to be our final one except for bioluminescence, which isn't actually a sensory structure. But our final receptors are photoreceptors, which detect light, hence the name, photo for light. So these involve light-sensitive pigments, molecules that absorb photons of light. As they absorb light, they undergo a chemical change, a change in conformation, that usually triggers a cascade of chemical changes that ultimately triggers an action potential and sends the information to the brain, if they have a brain. Again, photoreceptors can vary quite a bit in their complexity and structure. The simplest of these are called eye spots. So they can actually be even subcellular structures. So in protozoans, for example, they have an eye spot, or sometimes called a stigmata, which is just a small region that contains a bunch of photosensitive molecules, that can detect light. In multicellular animals, the eye spots generally are made up of multiple photosensitive cells. If you just have a spot of cells or pigment, all you can do is basically tell light from dark. So you can't determine images. You often can't even determine where the direction the light is coming from, since if you're transparent, that light can stimulate that eye spot from pretty much all directions. So if you want to avoid light, you swim until you're in a direction that makes your eye spot less stimulated. If you want to get to light, if you want to photosynthesize, like in Glina, You just kind of swim around the and then head in the direction where the stimulation is the strongest, basically. But if you're not moving, you can't really tell where the light is coming from. From there, photoreceptors can be increasingly complex. Ocelli are basically small, little, simple eyes. They're multicellular, light-sensitive structures, typically cup or bowl shaped, so they're shaped like a bowl. The advantage here is that if you're not just a flat surface, you have a cup shape, then light coming from one direction is going to impact one side more than the other. So while you may not be able to form images, you can at least tell the direction of the light and the intensity of that light better than if you had just a simple eye spot. And then true eyes are those that can actually form images that can focus that light into forming some form of images. Typically lenses are used, but there are actually some exceptions. So for example, Nautilus have what are called pinhole camera type eyes, where you just have a very small, you have a retina, the sensory surface is basically in a circular structure like our eyes, but instead of having a lens in front, you just have a very small opening. But that actually works the same way, which focuses the light onto the retina, so they can form images. You just need a lot of light to do it. Those with lenses can be broken down into compound eyes and camera type eyes. A compound eye has basically multiple units. Each unit has its own separate individual lens, but then the brain kind of combines all that information to perceive a single environment. In contrast, camera type eyes have a single retina and a single lens that can form images. And our final, and I know this is definitely the final slide, is I just briefly mention bioluminescence, because again, it's pretty widespread amongst the animal kingdom. Bioluminescence is the production of light. So here we're not talking about sensory structures at all. We're talking about the production of light using various chemical processes. Most of the chemical processes involved are actually pretty efficient and generally produce little in the way of waste heat. So you can keep up a bunch of fireflies in a jar, and the jar is not going to get hot even if they're all flashing their light. So unlike our regular incandescent lights, they're pretty efficient there. The process involved is the oxidation of a set of comp molecules called luciferins. They are oxidized by an enzyme called luciferase, and as that oxygen takes place, that process releases light rather than heat and light. And different versions of luciferins can produce different colors, so you can have red and yellows and green lights and so on. This process has evolved multiple times. In many animals, it may actually be carried out by endosymbiotic bacteria, but it also has evolved independently within animals. So it's found, for example, among protists and dinoflagellates, which can literally glow. So you can have basically glowing seawater if you have a larger number of dinoflagellates there. You flow your hand through it and you get these glowy bits as they are disturbed and they produce light. Some cnidarians can produce light. Cone jellies commonly do so. Some annelid worms, some mollusks, some arthropods, some echinoderms, various tunicates all have that capability. Typically found in deep sea animals where there is no natural light that they're competing with. So they can use that to attract mates, attract food, for example, by producing light, things like that. Some of them use light as a kind of an aposematic signal, a warning signal that they might be dangerous or poisonous, and so on. All right, so that is everything that's covered on the exam. Anybody have any questions so far? All right, so the exam, I haven't even really given it a lot of thought yet. It's been a busy week, and so I have something to do this weekend, not that I don't normally. But usually, what you should expect is sort of a mix of question types. So there will be some no Scantron is needed, so it will all be written not a big enough class to worry about Scantron. So just bring a writing stick, a pen or pencil, and that's all you need. There'll be multiple choice questions. There might be some matching questions. In fact, there's probably a good chance of some. You can think of just those last handful of slides could easily match, the sensory structure with what it does, kind of thing. There could be matching questions. There could be, and there could be fill in the blank questions. Maybe some true false, maybe not. There'll probably be one or two longer kind of questions. Not really essay questions in that you don't necessarily need to write out, a coherent essay format. But I don't care about paragraphs or things like that. But I might ask you to, describe, I don't know. I'm blanking right now, but you have some examples on the study guide. All right, where are we on time? Oh, we've got a tiny few minutes. We can probably run a little late here. I don't think there's any afterwards. I know where you got to be at 2, right? All right, so let's do a little bit of, so the first set of questions I'm going to go through are just the ones directly from the study guide here. So if you've looked at that, these are them, and these are actually old exam questions that I've used in the past. You might even see some again. So these are here mostly to give you a feel for my question writing style here, so you can kind of get to see the kinds of questions I might ask, because these are the kinds of questions I have actually asked. All right. So number 1, which of the following was not one of Darwin's contributions to science? I'm not going to read all the choices because otherwise we'll be here all day. So why don't you guys? How many say A, B, C, D? Okay, saw a few hands for C. Most of you are what? Are just going to leave that question blank? You can always take a guess, right? All right, so 2. Paraphyletic taxa are defined as groups of organisms. Add that. Which of these defines paraphyletic taxa? A, B, C, or D? Okay, got a little bit of mixed choices here. So paraphyletic taxa include the common ancestor of that group, but not all of the descendants. So reptiles, for example, would include the common ancestor of all living reptiles, but not all of the descendants if you don't include birds. So that is D. All right, number 3, compared to molecular data, using morphological data and cladistic analysis. I don't think we spent a lot of time on this, so this might be a little more guesswork here this time around. But compared to molecular data, using morphological data and cladistic analysis, the advantage that A, can be applied to fossil organisms. B, easier to produce data on large numbers of traits. Remember, talking about molecular data here. Traits are likely to be independent of one another, and homoplasies cannot be detected. All right. So how many guess A? How many guess B? All right. How many guess C? And D? All right. So molecular data, for the most part, cannot be applied to fossils. We generally don't. There are some exceptions, but for the most part, not. It is easier to produce data on large numbers of traits. That's true. And so it's easy. Nowadays, we can sequence fairly large chunks of genome and compare those to data that have been reported for other organisms and get pretty large molecular data sets. While you're limited with morphology, and it's much harder to categorize all of those anatomical features that you would need. Traits are likely to be independent of one another. That's actually kind of true for molecular data as well, isn't it? So that is actually an advantage. I think that might even be the best question here. And homoplasies cannot be detected. That is more of an issue with molecular data than with morphological data, where we might be able to detect homoplasies independent of the phylogeny. All right, so B is the one I was going for, but I think C might actually be acceptable as well. Anyway, Oh, no, wait a minute. I'm misreading the question, aren't I? Yeah, I'm an idiot. So don't do this, don't do what I'm doing. So, all right, so I got to parse the question here carefully a little bit. So it says compared to molecular, I was answering it from molecular data. So it's compared to molecular data, morphological data, including this advanced analysis, has the advantage of. So, may help to read it. I should read my own question. So morphological has the advantages that can be applied to fossil organisms. That's correct. It's generally much harder to produce morphological data. So B is actually not correct. So ignore what I just said. I don't even know. I'll be curious to see which one I marked when I marked this. Traits are likely to be independent of one another. Again, that's one I think we didn't really talk about this time because we didn't get into the statistics too much. But with morphological data, you have a much greater chance of non-independence. Different parts are connected to one another, so a change in one part will automatically mean a change in another. You have things like serial homology, so the same gene produces the same changes in different appendages, things like that. So there's a less independence, even if you might score them as separate structures, they're not really independent. And then homoplaces cannot be detected. We can in morphological data, so that's not really true. So the correct answer that I actually was going for is should have been A. So ignore that little check mark there. I misread this when I did this when I marked these off the other day, and started doing it the same today. All right. Which of the? Because once you misread it once, then you kind of get that in your head that that's what the question was going for. So. Which of the following is not a criticism of phylogenetic classification systems? Has the potential to increase excessive number of taxon names. That's true. Taxon names can be unstable to changing phylogenies. Then we're going to get into that a little too much, and I'm not sure I'd like to use that wording. But that is actually a potential issue. That is how you define a taxon, whether an organism falls into a particular group, depends on what part, on what the phylogeny is. You put that branch somewhere else, and it might, under certain definitions, fall out of that group. So it creates a certain degree of instability from the organism perspective. Eliminates paraphyletic taxa that are potentially useful for communication. That is true. So let me just think here. So this is a criticism. This is a criticism. This is a potential criticism. Yes, and does not accurately reflect a evolutionary relationship. So that's the benefit of phylogenetic classification. They do try to basically reflect evolutionary relationships, often at the cost of other things. So D should be the correct answer. Yes. All right. In the Linnaean classification system, classes are subdivided into what? Of those choices? Orders, C. Which type of symmetry is associated with a sessile or pelagic drifting lifestyle? Asymmetry, bilateral, or radial symmetry? Huh? All right, A. I'm going to say A. I'm going to say B. I'm going to say C. C is correct. Radial symmetry is basically, if you're sessile and not moving, you can, or you're just drifting around, you can interact with your environment in all directions. All right, based on the diagram on the right, which letter represents the most recent common ancestor of species 1 and 3? So what would be the most recent common ancestor? A, B, C, D. There we go. Yes, D. All right. Which of the following statements about invertebrates is true? A, they represent a monophyletic group comprised of 98% of all animal species, include species with a vertebral column, or include many single-cell species. I'm going to say A, B. Yeah, we'll stop there. All right. 9. The reproductive pattern in which individuals start out as female and later become male is known as. I'm going to say A. Protogeny, correct. The developmental process in which a hollow sphere invaginates to form the gut is called A. Blastulation, B. Cleavage, gastrulation, metamorphism, or schistocelia. I'm going to say A. B. C. Correct. Which of the following characteristics is generally true for species with direct development? Let me say A. Let me say B. How many say C? And how many say D? Yes, D is correct. All right. 12. Rigid skeletons derived from the ectoderm are known as A, B. How many say A, B, C, D, or E? All right, correct answer is exoskeletons. They're external, which means they're generally secreted by the ectoderm. All right, 13. Terrestrial invertebrates, such as arthropods and snails, secrete nitrogenous waste in the form of. How many say, so remember terrestrial here. How many say A, ammonia? B, uric acid? Correct, B is it. 14, as size, the length of an organism increases. How many say A, cell surface increases more rapidly than volume? How many say B, volume increases more rapidly than surface area? Or D, that increases at the same rate? Correct answer is B. Again, most of you haven't got that. All right, no, 15, blank, are long extensions of the cell used in locomotion and may have hair-like projections to increase area. All right, so how many say A, cilia, B, flagella, C, hydrostats, or D, pseudopods? All right, correct answer is flagella, which, so the keys here are long extensions of the cell and hair-like projections, which are found in, not all, but some flagella. So that kind of helps distinguish them from cilia. 16, what organelle is used by freshwater protozoans to regulate osmolarity and fluid levels? A, apical complex. B, contractile vacuum. Well, some of these you've seen in introbio, and a few of these you've seen elsewhere. C, Golgi apparatus. D, Malpighian tubule, or E, protonephridia. How many say A? You haven't seen that one yet, because we didn't cover that group in lab yet, or at all. B, contractile vacuole. C, Golgi apparatus. D, Malpighian tubule, or E, protonephridia. Not so many hands this time, huh? Not so sure. All right, well, if you want to guess, you should probably guess D. Guessing the others would be incorrect in this question. And by the way, so one of the sort of general things that people often tell you is that if you don't know the answer, you should guess B. It doesn't necessarily work for my tests. So the reason is, the idea is that if you're writing a bunch of questions, you don't generally want to put the one that you know is the right answer first, and so then you kind of come up with a false answer, then you write the right answer, and then you have to struggle to come up with a bunch more false answers. So people tend to, if they're just writing questions out, often there's a natural tendency to put the correct answer as B. One of the things I usually do is, you can see in this question, for example, I often alphabetize the answers. So kind of as a way of randomizing it, basically. All right, 16. What organelle is used by freshwater protozoans to regulate osmolarity and fluid? Oh, we just did that one, sorry. Yeah, that's also a B, and they're also in alphabetical order. All right, what type of sensory receptor detects the pull of gravity? I'm going to say A. So this is short-term memory here, guys. A, baroreceptor. B, chemoreceptor. C, georeceptor. There we go. All right, true or false, the top here. Homologous traits have a similar underlying structure because they were inherited from a common ancestor. I'm going to say true. I'm going to say false. Okay, so the non-voters are still out winning the voters there, so. And this you should be able to guess, you can guess on these easily. You got a 50-50 chance of getting it right. All right, the first cleavage during animal development is always along the animal-vegetal axis. So how many say true? How many say false? All right, it is true. But I really like true-false questions where I can use a word like always, because people often get thrown up by that, but sometimes it actually can be useful. In this case, it is. But sometimes people think, oh, it isn't always here. There's got to be an exception, so I'll write false. Not always. Sometimes there isn't an exception. So, and I like those. All right, 20. In deuterostome, the blastophore becomes a mouth. Is that true or false? The deutero second, stone mouth. So the mouth becomes second. The blastophore is always going to be the first opening, so that forms the anus of deuterostome. So that's false. Protostomes typically exhibit radial cleavage. Is that true or is that false? How many say true? How many say false? About 50-50. It's false. Protostomes typically have spiral cleavage. All right, matching. Deposit feeders. Which letter represents describes deposit feeders? A. Which letter represents suspension feeders? Anybody?
B.
B. Oops. Oh. Sorry. I thought that it had them going in order. I don't know why that came. I guess I didn't adjust the order here. All right. So I have no idea what's next. So I'm just going to click buttons. There we go. B. Suspension feeders. You got that right. And then by a process of elimination, you can do substrate feeders are D. Okay, fill in the blanks. There we go. Now you got to think up words. The outermost embryonic germ layer is called the ectoderm. Bioluminescence is produced when the enzyme blank oxidizes certain molecules. Luciferase. Lucifer, and then all enzymes end in ASE, so luciferase is the enzyme. All right, according to the principle of blank, the phylogenetic tree that requires the fewest evolutionary transitions is most likely to be correct. Parsimony. Blank release large, and there's actually two blanks there. So blank release large quantities of eggs and sperm directly into the water column. Broadcast spawners. And 27 species which it should really be in which individuals have both ovaries and testes are known as. And there are actually two answers here that you can that I would accept. Huh? Nope. Monoecious. Dioecious means there are two kinds. Monoecious means there's one kind that has both. So that kind of keeps that straight there. Or what's another word you can, instead of monoecious, we often use? Hermaphrodites or hermaphroditic. All right. Let's see, where are we on time? You've got, by my watch, about 6 minutes or so. You guys want to do more review, and we can just start. If we start lab, we might run lab. The lab might run later. I don't know if anybody needs to get out of lab, so I don't want to force you out of lab here. But since I know where you're going to be next, we can run a little longer here if you like. What do you guys? Any objections? I don't know. It should be a reasonably full lab. I mean, in the past, this was one that usually people finish on time, not like with a lot of extra time, but so how much do you want to go? How much extra can you go? It depends if you're one of the people who finishes fast. I don't remember if you finish relatively quickly or usually one of the last people there.
The last one.
Huh?
I'm not going to be off the last one.
Okay. Let's go for another 15 minutes and we'll go through some. All right. Well, let's see how some of these we've already done. I think before I took a little break at one point. The branch lengths of evolutionary trees may be used to illustrate. Anybody remember?
Nothing is one of them.
Nothing. Nothing. Yes. You remember? What else? Time and
evolutionary changes.
Yeah, amount of evolutionary changes, genetic distance, things like that. Number of evolutionary changes. All right, fill in the blanks. Blank is used to polarize traits as ancestral or derived. A what? Huh? Outgroup. The principle of blah, 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 we just did this one. Parsimony. Blank are traits that evolve for a particular function.
Adaptation.
Adaptations. Cephalization is associated with what kind of symmetry?
Bilateral.
Bilateral. All right, what are the main categories of Linnaean classification from most general to specific? Kingdom, followed by phylum, followed by class, followed by
order,
followed by
family,
and
genus,
genus,
species.